grade 9s, and welcome to lesson 1 in reproduction. Today we're going to talk a little bit about mitosis. Okay, so by the end of this video lesson and next days in class, you should be able to kind of illustrate and explain the process of mitosis or mitotic cell division and kind of observe and explain the dynamic nature, the idea that the cells are continuously in one phase of the cell cycle. So your body needs new cells throughout your life. Even if you've stopped growing, old cells will die, they'll get injured, and they need to repair. And different cells live for different amounts of time. Okay, so depending on what their function is and where they perform that function. So things like brain cells last a really long time and they don't typically regenerate, but other things like the lining of your stomach or your skin cells are kind of always being replaced. Hair cells grow really quickly as well. And other ones last for kind of a few months to a few years. So the cell cycle consists of 3 distinct phases. Okay? So interphase, that's the growth phase. So if you look at this diagram here, it's this growth phase here. So interphase is when the cell's just living and doing its job and growing. And then mitosis is when the cell is preparing to divide. Okay, so that's this piece of the puzzle here. And these are the phases that we're going to learn about over the next few days, the ones with the little red star beside them. And then the process when the cell actually divides into 2 new cells is called cytokinesis or cell division. So terms you need to know. So you need to know that interphase is the cell just kind of living its life and doing its job. It's the cell carrying out its daily functions and duties. At the very end of interphase, if the conditions are just right, the chromosomes will replicate in preparation for cell division. So mitosis is when we're actually copying that genetic material and then separating it into 2 cells. So we take that copied genetic material, that was copied right at the end of interphase, and then we split it into 2 identical sets of chromosomes or 2 identical cells. And again, cytokinesis is when the cytoplasm with all those organelles you talked about in grade 8 science divides into 2. So chromosomes are double-stranded, tightly coiled DNA or chromatin that carries that DNA, that genetic material. And your genetic material is responsible for producing new cells with those same characteristics. So there are several hundred trillion cells in the adult body. You started out as 1 cell, and that 1 cell divided into 2 cells. Those 2 cells divided into 2 cells, so you ended up making 4, and so on and so forth. And we're going to talk about that process of reproduction and how we got from that single cell to the multicellular organism you are now. Okay, but basically, the short answer is by having that cell divide, by cell division. Okay, if division only happened, then each cell would only have part of the nucleus. So every time you divided, you'd have half the information. So we have to make a photocopy, basically, of our genetic material, and then we have to split it equally into those 2 cells. And that process is called mitosis. So mitosis occurs in a set of series or stages, okay? So interphase is that growth phase, that growth and replication, but the phases of mitosis, mitosis, sorry, are often abbreviated PMAT. Okay, and that's P for prophase, M for metaphase, A for anaphase, and T for telophase or telophase. And then that last step is when we actually divide the cell into 2. So basically, we have interphase, that's for growth, and your chromosomes copy. Then we have mitosis. Basically, that divides the nucleus. That's the brain of the cell. So the nucleus with all that genetic information. And then that cytokinesis divides up the rest of the cell. And so we take 1 cell that's got 2 copies of the chromosomes, and we divide it into 2 identical cells. Okay, so you might want to think up an acronym for these stages. You can use that one, PMAT, if you like, or you can come up with another one. I for interphase, P for PMAT for prophase, P myself, so metaphase, at anaphase, 10, telophase, and then that o'clock or clock for cell division. So go ahead and think of your own if you'd like, or just use one of those, but you need to remember at the very least PMAT and what those stand for. Okay. So in interphase, this, oops, goodness. No ending show, no ending show. Just go away. All right. No, it's not going to go away. I'm going to go previous, and then I'll come back to this. There we go. So sorry about that. So this occurs, interphase occurs right before mitosis. Okay. And so it gets ready for mitosis by doubling the amount of chromosomes. So if I was a highlighter, I'd be, kind of, okay. So the chromosomes get replicated. So basically, we don't have twice the genetic information. We just have a photocopy of the stuff we already have. So if I gave you 2 copies of the notes for the reproduction unit, you're not twice as smart if you go through both of those. You just have 2 copies of the same thing. So at the end of interphase, the chromosomes are replicated to prepare. We make a photocopy of them to prepare for cell division. So that initial cell we call the parent cell, and it duplicates its chromosomes. So chromosomes appear as thin thread-like coils. So when the cell is just hanging out doing its job, so if we draw this in, there's the nucleus, your chromosomes are all unwound, and they look like thread off the spool. Okay? So each chromosome, again, starts to kind of copy. So this is where the language is a little tricky. This is a chromosome. This is a single-stranded chromosome. And then this is a double-stranded chromosome here. Again, it's made that photocopy. So they're both chromosomes. Sometimes we'll use the word sister chromatids. This chromosome is double-stranded. It's got 2 sister chromatids. Okay? So that's interphase. So if we were trying to identify a picture of interphase, you'd be looking for something where kind of all the DNA is kind of unwound. Prophase is the first stage. So in this one, oh, we're still going here. I just had to check. I don't know where that thing went. Sorry, I just had to make sure that I was still recording here. So prophase is the first stage of mitosis. And what we're going to see here is the big thing is we're going to see the chromosomes start to condense. So those chromosomes are going to start to wind up into little packages. Okay, so you can see they start to kind of condense here in this picture over here. But in animal cells, we're also going to see these centrioles forming, okay, and spindle fibers forming. So this is basically going to go and attach to those chromosomes eventually and be like a rope to pull it back. You're also going to see this nuclear membrane start to disappear. So depending on what part it's in, this might kind of appear as a dotted line as the nucleus starts to kind of break down. The next phase is called metaphase, and this is the one that students usually kind of find it easy. This is basically where all the chromosomes line up in the middle. So basically meta means middle. Okay, so meta is middle. Okay, and so the nuclear membrane will be totally gone, and you'll see the chromosomes lined up in the middle. You'll see the centrioles at the end that I'm drawing now, and then you'll see these spindle fibers, these ropes that are going to go and attach to the chromosomes, kind of coming out to the center. Okay, so again, metaphase, all the chromosomes are lined up in the middle of the cell. Then these spindle fibers will start to attach to what's called the centromere of these chromosomes, and it'll pull those sister chromatids apart. And they were photocopies. So those sister chromatids were just the photocopies, so it'll start to kind of pull those photocopies apart. So these spindle fibers will start to shorten, and these chromosomes will start to kind of separate and be pulled to either side of the cell. Okay, so this is, you're going to see a picture where they're starting to move to opposite sets of the cell. Okay, in telophase, what you're going to see is you're going to start to see two new nuclei forming. So those chromosomes would be kind of on either side of the cell. You'll see the nucleus, that nuclear membrane starting to reform, and you'll see the cell start to pinch in a little bit here. Plants and animal cells work a little differently. Animal cells, because they don't have a cell wall, they'll just simply pinch in and kind of pinch into two. Plant cells have to build what's called a cell plate to make that cell wall. And this is basically, telophase is the last stage of mitosis. So mitosis ends, and then the only thing left is to actually split the cell into two identical pieces. So this is cell division or cytokinesis. So we split that cell and you end up with two identical cells, each with their own nuclei or nucleus. Okay, so again, the nucleus would be reformed and that DNA would start to unwind again. Okay, and these are exact copies of their original. So we end up with, we call these two daughter cells, and these daughter cells are identical to each other, and they're identical to the parent cell. Okay, so the whole process of mitosis takes about 20 hours in mammals, but can be as quick as 20 minutes in bacteria. So now that you've watched the video, what I'd like you to do before you come back next class is take a few minutes and try tasks 1 and 2 on page 7 to 9 in your booklet. So basically, you're going to look back and review those notes on mitosis, and you're going to try to identify the stages and number them in terms of which order they come from. So give it a go, and you'll have a little bit more time in class, but I'd like you to have a try at home, and then we'll practice more when you come to class. Thanks for watching.